Hi, welcome to exam 4 written portion. This is done by Katie Jones. Hope you enjoy it. Alright, so question 1. Below are the lac operon genotypes of certain E. coli strains, and these spaces below indicate whether the lac operon will be inducible, non-inducible, or constitutive. Assume that the glucose levels are low in all cases. For the partial diploid strains, part D and E, describe the overall phenotype of the cell. Do not provide a separate answer for each copy of the lac operon in the cell. Note that IS is dominant to I+, I+ is dominant to, L. LS is dominant to L+, L+ is dominant to L-, and OC is dominant to O+. So 1A. So we have L-, P+, O+, Z+, Y+, and A+, and is non-inducible. 1B. We have IS, P+, O+, Z+, Y+, and A+, and is constitutive. 1C. We have L+, P-, OC, Z+, Y+, and A+, and it's non-inducible. 1D. We have on the F' plasmid, we have IS, P+, O+, Z+, Y+, and A+. And on the chromosome, we have L+, P+, OC, Z+, Y+, and A+. Lac L is always on. Operator sequence doesn't work. Proteins will not bind to it, essentially. And Lac Z, Lac Y, and Lac A are operational, and as constitutive. 1E. L+. On the F' plasmid, we have L+, P+, O+, Z-, Y+, and A+. And on the chromosome, we have L-, P+, O+, Z+, Y+, and A+. And Lac L is operational. Proteins will bind to Lac O. Promoter sequence allows proteins to bind to it. Lac Z, Y, and A are functional, and it's inducible. So question 2. After DNA sequencing using the Sanger method described in class, the DNA fragments were separated by electrophoresis on a gel. What would the bonding pattern look like on the gel? So what we have here is a picture of the strain and then what it will look like from the gel. So question 3, A and B. So 2 mRNA codons have the sequences indicated below in parts A and B. Consider the Wobble rules and give all possible tRNA anticodons that can bind to the following codons, and be sure to indicate the polarity of each anticodon. So for 3A, we have, which was 5' GCC 3'. We have 3' CGG 5', 3' CGA 5', 3' CGI 5', and B, we have 5' AAG 3', and so we have 3' UUC 5', 3' UUU 5', 3' UUA 5', and 3' UUI 5'. And so C and D, so we have 2 tRNA anticodons have the sequences indicated below in parts C and D. Consider the Wobble rules and give all possible mRNA codons that combine to the following anticodons, and be sure to indicate the polarity of each codon. So for C, we have 5' UAU 3'. So we have 3' AUG 5', 3' AUA 5', 3' AUU 5', and that's it. And for D, it's 5' AUG 3', and so we have 3' UAC 5', and 3' UAU 5'. So for our last question, question 4, the average mass of the 20 common amino acids is about 125 daltons each. Suppose an E. coli protein called your favorite protein, as known YFP for short, is 75 kilodaltons. So for A, what is the length of the nucleotides of the protein portion of the E. coli mRNA, the coding region that is translated to make YFP? And so we break it down to convert the kilodaltons into daltons, and then the number of amino acids. So the number of daltons is 600, and then converting that number of daltons into the number of amino acids, we get 4.8, and since you can't really have 0.8 amino acids, we're just gonna kind of round it to 5 amino acids. And then since there are 3 bases per amino acid, there's 5 multiplied by 3 is 15 nucleotides. 4b. The E. coli mRNA that makes YFP contains a 5' UTR and 3' UTR. What's the function of the 5' UTR and 3' UTR? And it's the 5' UTR and 3' UTR are like stop codons. They're found downstream of the stop codon and force translation to stop just in case stop codon is defective. Like you have a deletion in that codon, or an addition, or a duplicate, or, the number of many things that could cause that to be that way. Oh, flip. 4c. How many ATP molecules are required to translate 100 copies of YFP? And it's 5 ATP per 1 gene, so 100 times 5 is 500 ATP. And 40, how many GTP molecules are required to translate 50 copies of YFP? And it's 15 GTP per 1 gene, and so that's 15 times 50 is 150 GTP. Thanks for watching, and have a wonderful day.